Hello everyone, welcome to Merrimack STEM Academy. Today we're going to give you the rundown on everything you need to know about mutations. Mutations are changes in DNA caused by an error. To understand mutations, you need to understand central dogma, so if you haven't seen our central dogma video, please check it out. There will be a link in the description. Mutations are important because changes in DNA can lead to harmful effects and cause many genetic disorders you may have heard of, like hemophilia and sickle cell anemia. So without further ado, let's get into it. We are going to start by talking about the two main causes of mutations. First, mutations can be caused by random error. There are many mechanisms of checking DNA to make sure it is copied correctly, but so much DNA is replicated in our bodies daily, and occasionally there can be errors. Mutations can also be caused by substances that harm DNA, called mutagens, such as radiation and certain chemicals. For example, UV light and sunlight can damage DNA and lead to tumors and skin cancer. Be sure to wear sunscreen to limit your exposure to UV light and protect yourself. There are many different types of mutations. The first type is called a silent mutation because it has no effect on the amino acid sequence, so it's silent. In a silent mutation, a DNA nucleotide is changed, but the resulting codon still codes for the same amino acid, so the amino acid sequence stays the same. The next type is a missense mutation. This occurs when a DNA base is changed, but this time, instead of coding for the same amino acid, the amino acid is different. Nonsense mutations occur when a nucleotide is changed to code for a stop codon instead of an amino acid. As a result, translation ends earlier than it is supposed to, and the amino acid sequence will be incomplete. There are also insertions and deletions, which are pretty self-explanatory. A nucleotide is either added to the DNA sequence or deleted from the DNA sequence. Insertions and deletions in the coding region can change the reading frame by changing all the codons after the mutation. As a result, the DNA sequence is drastically changed and will most likely code for a very different amino acid sequence than it is supposed to. The types of mutations are categorized into two groups. Silent, missense, and nonsense mutations are called point mutations because the DNA is only changed at that one point. The reading frame stays the same. Insertions and deletions are called frameshift mutations because the reading frame will change. Frameshift mutations can be very harmful because they can change large parts of the amino acid sequence. Now that we know what mutations are and the different types of mutations, let's talk about what mutations actually do by tracing the path of a mutation. Mutated DNA is transcribed to create mutated mRNA, which can produce a protein with a different amino acid sequence. The amino acid sequence can be affected in a few different ways. First, it's possible that the amino acid sequence stays the same. Silent mutations don't affect the amino acid sequence. It is also possible that the mutation occurred in the untranslated region of DNA rather than the coding region, in which case the protein may stay the same. Another possible outcome is that the amino acid sequence is changed because of the mutations. A missense mutation would cause one amino acid in the sequence to change. A frameshift mutation would also cause many amino acids in the sequence to change. Another outcome is that the amino acid sequence has a different length. A nonsense mutation produces an incomplete amino acid sequence because of the early stop codon. Insertions and deletions will often change the amino acid sequence's length. The shift in the reading frame can change the location of the stop codon. Alright, let's say that the amino acid sequence is altered in some way. How does this affect the protein as a whole? Well, protein structure is determined by interactions between amino acids. So, if the amino acids are changed, their interactions are changed, and the protein structure can change as well. The amino acids' interactions are affected by the amino acid's size and its chemistry, if it's charged, polar, or nonpolar. If the mutation causes a bigger change in the amino acid's size and chemistry, then the protein structure is more likely to change. As you may have heard, structure leads to function, so changing the protein structure may affect its ability to do its job. It is possible that the protein structure doesn't change that much and the protein can still function as normal. Another possibility is that the protein will function much better than before. This is unlikely, but this mutation could lead to natural selection. Most likely, the mutation will cause the protein to be less functional or non-functional. This could cause a genetic disease. In this case, the protein may be harmful to the cell, or it may be degraded by the cell because it doesn't function. Let's review what we've just talked about. Point mutations affect one nucleotide and don't change the reading frame. Silent mutations produce the same amino acid sequence. Missense mutations change one amino acid, and nonsense mutations result in an early stop codon. Frameshift mutations affect the reading frame, changing all the amino acids after the insertion or deletion. A change in the DNA may change the amino acid sequence. The interactions between R groups change, so the protein structure may change as well. Changing the protein structure usually changes its function. Alright, let's see what you've learned and put your knowledge to the test. For the list of mutations below, pick which one is more likely to produce a less functional or non-functional protein. Pause the video here and see if you can figure it out. Okay, let's see how well you did. First, silent mutation versus missense mutation. Well, silent mutations don't affect the amino acid sequence. A missense mutation does change an amino acid, which could affect protein structure and function, so the answer here is missense mutation. Next, a missense mutation that affects the ligand binding site, or the first amino acid of the protein. The ligand binding site needs to have a certain structure so the ligand can bind, like having a key fitting into a lock. Disrupting the binding site could prevent the ligand from binding, whereas changing the first amino acid probably won't affect the ligand binding. So, most likely, the binding site mutation will be more harmful. Third, a nonsense mutation versus a missense mutation. Well, a nonsense mutation causes an early stop, so the amino acid sequence is incomplete. A missense mutation affects only one amino acid, but most of the sequence will stay the same. So, the nonsense mutation will probably be more harmful. Moving on, a nonsense mutation at the 3rd codon or at the 100th codon. Well, for the nonsense mutation at the 3rd codon, there will be 2 amino acids before translation is stopped. For a nonsense mutation at the 100th codon, the protein will have 99 amino acids. 2 amino acids are not a complete functional protein, so this protein is more likely to be non-functional. Insertion versus deletion? Well, this is actually a trick question. Sorry. Both will result in a shift in the reading frame, changing all the amino acids after the mutation. It depends on the specific situation, not whether the mutation is an insertion or deletion. Let's look at one of those specific situations, an insertion at the 100th codon or a deletion at the 10th codon. What's important to look at here is the location of the mutation. When the frameshift occurs earlier in the sequence, it will affect more amino acids, causing the protein structure to change more. So, the frameshift at the 10th codon is more harmful. Now we have a question about a specific amino acid change, from arginine to lysine, or arginine to glutamic acid. Arginine is a positive amino acid. Lysine is also positive, whereas glutamic acid is negative. When looking at amino acid changes, we want to think about how the change affects the size and chemistry of the amino acid. In this case, lysine has a similar size and the same chemistry as arginine. Both are positive. This would not significantly change the interactions between R groups. Glutamic acid has a different chemistry because it is negatively charged. This is a big change, so it will probably affect the interactions between R groups. For example, arginine may have formed an ionic bond with a negative amino acid, but glutamic acid would repel this negative charge, so the switch to glutamic acid is more likely to be harmful. Last question, we're going to look at another amino acid change, from valine to tryptophan or alanine. We're going to use the same process, looking at the amino acid size and chemistry. All of these amino acids are nonpolar, so the chemistry is not changing. However, we can see that the size of the amino acid is changing. Valine and alanine have similar sizes. However, tryptophan is much larger with 2 ring structures. The size of tryptophan may disrupt the protein structure, so the change to tryptophan is more likely to be detrimental to the protein structure. Here is a summary of the answers. Let us know in the comments how well you did. And on that note, you've learned everything you need to know about mutations. Thank you for listening. Have any questions or video ideas? Comment them below. And be sure to like and subscribe for more videos about biology, chemistry, and everything STEM related.